Oh, so how are you doing?
Hey, how's it going?
Doing good. What's up, Cam? How are you doing?
Hey, I'm doing good. How are you?
I'm doing good. So wait for a few minutes, see if anyone else comes. What's up, Krishal? How are you doing?
What's up, Sid? Good, how are you?
I'm doing good. All right. Go ahead and get started, and then if anyone else joins, we can just pause. But so today we're gonna be looking at like a biochemistry passage from the bio/biochem section of the MCAT. And the passage is called Toxic Therapeutics. So today is going to be a bit different in that we're going to look at the passage before we look at the background, because the background for the passage is very directly tied to the passage, and like specifically the experiment in the passage. So it's a bit hard to explain the background without knowing the experiment that we're talking about, because it's a background for that experiment. So we're going to look at the experiment for it first and then figure out how to walk through it using the PowerPoint. So let me switch to the passage. Awesome. So can everyone see the screen? All right. So the passage says that the active metabolites of 5-fluorouracil produce cytotoxic effects by inhibiting thymidylate synthase, an important enzyme for nucleotide synthesis. And then it shows us that reaction. So so that's the reaction here. So could someone read the second paragraph? Sorry.
Yeah, I can read it. Starting at like many.
Yeah, sure.
Like many chemotherapeutics, 5-FU dosage is limited by its toxicity and the damage it can cause to the body. Because of this, researchers were interested in developing a chemotherapeutic that is non-toxic to non-cancerous cells. They decided to investigate an analog of F-5FU, 5-fluorocytosine, as a prodrug in conjunction with a fusion protein composed of cytosine deaminase, CD, bound to annexin V protein via disulfide linkages. Free annexin V is known to bind phosphatidylserine, which is primarily expressed on the surface of tumor cells. They hypothesized that if the CDAV complex localizes only to cancerous cells, the 5-FC prodrug can be injected intravenously and converted into its active metabolite via CD.
Awesome. And then if someone could read the next paragraph.
To see the affinity of CDAV to cancerous tissue, researchers perform CDAV binding and stability assays on different cancerous tissue types: HAAE1, MCF7, MGA-MB231. First, researchers obtained specific binding data between biotinylated CDAV to phosphatidylserine targets on each tissue type, and then determine each dissociation constant, KD. The observed KD values are presented in Table 1.
Awesome. And then if someone could read the last paragraph.
This data confirmed a strong binding association between CDAV and phosphatidylserine. Next, the researchers determined the viability of MCF7 cells at 36 and 9 days post-treatment when treated with 5-FC only, 5-FU only, or CDAV and 5-FC. The experiment was repeated in triplicate using 0, 500, or 1000 micromolar concentrations of 5-FC/5-FU.
Awesome. So just from reading the passage, what are your takeaways from that? Like, how are you feeling about it?
Can you scroll down again for a second?
Oh, that's okay. Is it like, is it hard? Is it easy? Is it confusing or is it simple?
It was a little confusing. I'm like,
Hmm.
right now I kind of get that 5-FC is like a chemotherapeutic
Mm,
type of drug for cancer.
yeah.
Um, it is somehow related to this thing called CDAV,
Hmm.
and then the CDAV thing is um, trying to bind to phosphatidylserine, which I think is part of a cell membrane.
Yeah.
Um, and they're trying to see like how well does it bind to different tissues.
Yeah. I mean, yeah, that's a really good start. So that's kind of what we're dealing with, but I'll admit that like this passage is pretty complex. So we're gonna walk through this and try to make it pretty simple. So you should feeling pretty comfortable about it. So let me stop screen sharing here and let's go to the PowerPoint. Awesome. Okay. So let's start off here from the very beginning. So we kind of want to figure out what 5FU does. So the main subject of our passage is 5FU. And so can someone tell me what a nucleotide is? So a nucleotide. Uh, just what a nucleotide is. Yeah.
Um, phosphate plus base plus sugar.
Yeah. So those are the components of a nucleotide, and a nucleotide is what makes up DNA. So every cell has DNA, and DNA is made up from the building block, which is a nucleotide. So nucleotides are the building blocks of DNA. And so an important component of DNA is a nitrogenous base. So it's the part in purple right here, a nitrogenous base. And so it's called a nitrogenous base because you have nitrogen in it. But to make DNA, we need all sorts of different nucleotides, right? And how nucleotides differ is that they differ in their nitrogenous base. So we want all sorts of nitrogenous bases and different nucleotides. And to create all the nitrogenous bases and all the nucleotides that we need to convert between them. So one of the important enzymes is thymidylate synthase. So can everyone see this part I'm circling right here? My mouse.
Yes.
Okay. So we want to use this enzyme to convert between different nucleotides, right? Because we want to have a good balance of all the nucleotides to make our DNA. And if we don't have a good balance or abundance of all our nucleotides, we can't properly replicate the DNA and the cell is not going to do very well. It's most likely going to die. So let's say you had a cancer cell, right? And you wanted to kill that cancer cell. What would you want to do to this enzyme that converts uridine to thymidine and gives us the necessary nucleotides we need? What would you want to do to that enzyme if you wanted to kill that cancer cell?
Cleave it.
Right. So you would want to inhibit that enzyme. So you would want to stop its function. So yeah, cleaving it could be that is a good example. But here what they just call it, usually in general they'll just call it an inhibitor. So 5FU is our inhibitor. So it's going to bind to this thymidylate synthase enzyme. So it's an enzyme because it ends in ASE. So all enzymes are going to end in ASE usually. So it's going to bind to it and stop us from converting uridine to thymidine. So it's going to stop us from having all the nucleotides we need to replicate DNA. And that is going to help us kill cancer cells. So that's great, right? That's what we want. But there's a caveat. So here is a diagram where you can kind of think about what that means. So it's great, right? It binds to cancer cells and it inhibits the enzyme. So we don't make all the nucleotides we need to replicate DNA. So that's good. But on the other hand, it can also bind to our healthy cells, right? And if it binds to our healthy cells, we don't want to stop replicating DNA in our healthy cells, right? We want those healthy cells to live. So in that case, that would be toxic. So that's why, even though it can help cure like tumors and stuff like that, it can still be toxic to your healthy cells. So that begs the question, how could we make something that is only going to bind to our cancer cells and dodge our healthy cells? So it's going to avoid our healthy cells. Okay. So that's the whole point of this passage. And so the thing you're starting out with, the toxic therapeutic, is 5-FU. So we want to come up with a clever way to make another version of 5-FU that does not target our healthy cells. Okay. So how we do that is they have an innovative way called a pro-drug. Okay. And this is where you take a drug and you bind it to another protein called a carrier. And so when that drug and carrier come together, they form this thing called a prodrug. Okay. And so this prodrug carrier complex, it's only going to bind to specific cancer cells. So it's not going to bind to all the cells. It's only going to bind to the specific cells that we want to bind to. Okay. And so in this experiment, what they're doing is, instead of using 5-FU, which we know binds to like almost every cell, we're going to use an analog. So an analog is something that is very similar, but has maybe like a slight alteration. So that analog of 5-FU is going to be called 5-FC. So we're going to bind 5-FC to a carrier protein. When 5-FC is bound to that carrier protein, it's known as a prodrug. So that 5-FC prodrug is going to go and bind to the cancer cells. And only after it's binded to a cancer cell, it's going to be converted back to the 5-FU drug, so the active form. So that way it's only acting on the cancer cells. Okay. So can someone explain what a prodrug is?
Okay, I want to say, okay.
Yeah.
Prodrug is a complex between the actual drug and a carrier.
Right.
A carrier protein. This prodrug goes to cells and does it bind to them and then releases the actual drug?
Yeah, exactly. Yeah.
I have a question. How does the carrier differentiate between the regular drugs and or the regular cells and the cancerous cells?
So that is actually a great question. So it'll be on the next. So in the next slide, we're going to talk about that.
Okay.
So going to Cam's question. So remember we talked about how we have 5-FC, which is different from 5-FU. It's very similar, but it's slightly different. So it's an analog. So it binds to, so our carrier protein is this thing called cytosine deaminase bound to a Nexin V protein. So you ask Cam, how does it bind to the cells? Well, in our carrier protein, so our carrier protein is made up of CD and AB. AB binds to phosphoatidylserine, which is a phospholipid. So phospholipid is what makes up a cell membrane. So AB binds to phospholipids. So that's how our carrier protein binds to the cells. Does that make sense?
Yeah, that makes sense.
And so our carrier complex, which is what I'm circling right now, the CD and AB, binds to 5-FC and it delivers it to the cancer cells, right? So they want to make sure, what the experimenters want to test is how strongly the CD-AB complex binds to different cancer tissue types. Because they want to know whether that CD-AB complex is actually going to bind to the cancer cell so it can deliver 5-FC there, where 5-FC is going to be converted back to 5-FU and its active form can carry out its effects of stopping DNA replication.
Okay, so if I were to summarize that,
yeah,
I would say that the thing that finds, that is like that locates the cancerous cells. The
Mhm.
differentiation is by this the CDAV complex is what we are really trying to determine if it actually binds to cancerous cells or if it's has this like global binding.
Exactly. Yeah.
Okay, and then we use the drug to bind to the complex
Mhm.
that should be on the cancerous cells only.
Right.
Okay, that makes sense.
And so then my question is, if we don't add any CDAV complex with the 5FC, is it going to be converted to the active? So, okay, so another thing is within the CDAV complex is what converts the 5FC back to the 5FU after it's bound to the cancer cell. So once this whole thing binds to the cancer cell, the CD is going to convert the 5FC back to 5FU. So the active form is present now. So if you add 5FC alone, but you don't add any of the CDAV complex, is it going to have any effect on the cancer cells? Right. It's not going to have any effect because you don't have any CD within the CDAV complex to convert the 5FC to the 5FU form. Okay.
The FC has no effect on if it was just in like a
Yeah, exactly.
okay.
So
It's not like FU. FU would kill everything. FC
Yeah.
would do nothing. Okay.
So the whole point, if you think about it, is like we want to put the inactive form and send the inactive form to the cells we want dead, and then only when the inactive form is at those cells will we convert it to the active form so it can start acting. So we're kind of choosing which cells we want to send it to, and we choose which cells based on the CDAV complex. Okay.
Sid, I had a question.
Yeah.
So the CDAB complex that's present on the phospholipid bilayer, or
Right. So you add the CDAV complex, but it binds to the phospholipid bilayer.
okay.
So
So then how come it doesn't? How come like 5FC isn't converted to 5FU when it's not attached to the phospholipid bilayer?
I think it only acts when it only gets converted after binding to the phospholipid membrane. So I
Okay.
think that's just something you have to like assume.
Okay.
Yeah. So because so actually how you want to think about it is first the CDAV complex is going to bind to the cell membrane, then the 5FC is going to locate the CDAV, and then it'll be converted to the 5FU. So kind of assume the CDAV is already goes and binds here, and then the 5FC comes and finds it, and then gets converted to 5FU. Okay. So, okay, next technique we want to talk about is something called an immunoblot. So an immunoblot uses something called an antibody. So an antibody is it's a component of our immune system, and you can also make them in a lab. And so what they do is they bind a specific protein called an antigen. So you can use them to detect stuff in a cell, and you can also use them to detect if a cell is viable. And so how you can do that is if a cell is viable, it will still have proteins in it, and you can detect the cell by using antibodies that are anti to it. So when an antibody is specific for something, you called it an antibody. So if you want to detect whether, for example, cell X is viable, you would use an anti-X antibody. Okay. And so why that is relevant here is because we can detect whether the CDAV complex with 5C actually has any effect on cancer cells by detecting if the cells are viable after we send that complex in to try to kill the cells. So that is why we're using an immunoblot. So can someone tell me the purpose of an immunoblot in this case?
It's to determine if the antibodies are viable or not.
So we're trying to determine if the cells are viable.
Oh, okay.
So it's
Yeah. Maybe we're trying to see if this CDAB complex will actually convert FC to FU, and if that conversion will actually kill the cells that we need to kill.
Yeah, exactly.
Okay.
So
What type of antigen is it looking to bind to? Like what exactly is the antigen in a viable cell?
So the antigen is basically in this case, in this experiment, it's just going to be any protein and the cell that we're trying to look at. So they'll say they did an immunoblot for these X cells. And so if a band shows up on the immunoblot, like right here, that means the cell is still viable because it's detecting stuff within that cell. So we don't actually know which protein it is in this case, but all it is telling us is that that cell is still viable.
This might be a silly question, but once a cell dies,
Yeah.
all of this proteins and the things within it will also die.
Yeah, so they'll probably be released into like the outside and they might be degraded, for example. So if a cell dies, you would not expect to see the bands right here.
Okay.
Yeah, so for this experiment at least.
Wait, so why would the cell die if the proteins are not binding?
So we're trying to detect the innate proteins within that cell. So if our 5FC CDAB complex binds to the cancer cell, theoretically that they're going to kill that cell, right?
Oh,
So
okay.
if it kills that cell, all the proteins are going to be released and like probably degraded somewhere. So the antibodies are not going to be able to detect those proteins within the cell we're looking at. So that cell would not show up. Those bands would not show up because the cell would be dead.
Okay, so we can only use this for like for interactions that would cause like a cell to die, right? We couldn't like it's like a
No, so you could use it for any cell. We're just trying to see if the cell is still living after. So think of this as like we're just testing out this complex right here. We're trying to see if this thing is going to do anything to the cell. But we cannot send this complex in and just check the cell normally, and we would have bands show up because there would be proteins within that cell. But we're trying to see if this complex is going to kill the cell and cause those proteins within the cell to be undetectable because the cell would be dead. So the only reason why we're using an immunoblot in this case is to measure if the cell is living or not.
Oh, okay. So
So you could use this in any case normally, because normally you would expect the cell to be alive, the proteins would be within it, and you would get bands on the immunoblot.
so the actual antibodies and antigens doesn't matter. It's just like if the
No,
cell
yes, the antibody here is just a detector,
Okay.
whereas this thing right here, this is the like compound we're testing out to see whether it has an effect on the cell.
Okay, that makes sense.
Okay, and so another thing you need to learn about is something called a dissociation constant. So a dissociation constant can be used to figure out how much a ligand dissociates from a protein. If you have a lot of, if you have less dissociation, that means there's the protein and ligand are bound together more of the time. There's strong affinity between the protein and the ligand if you have less dissociation. So if you have strong binding between a protein and a ligand, are you going to have a low KD or a high KD?
Low KD.
Yeah, exactly. And so for our 5-FC-CD-AB complex, we want the lowest KD possible between that and the cancer cells because we want them to bind strongly to the cancer cells so they can have an effect. And so this chart right here is a review from I think our first session. So these are some different classes of enzymes, and a particular one we want to focus on here is transferase. So can anyone tell me what a transferase does?
It transfers functional groups from one molecule to another.
Yeah. So one example of functional grouping and transfer is a methyl group. So a CH3 group. So basically transferase is an enzyme that transfers stuff. So it's the same thing as what its name suggests. Okay. And this one is also a review from previously. So can someone tell me what a cofactor is?
It's an inorganic chemical or molecule that helps the enzyme do its function.
Exactly. Yeah. And so some examples are magnesium, zinc, iron. So like those are all metal ions, and they bind to an enzyme and they help them do its job. And you can also have something called a coenzyme, which is an organic group. So that is usually going to be like FAD or NAD. So it's like usually going to be a carbon molecule, not like just a metal ion. So we've gone through all the background that we need to know for the passage. So you hopefully should have a better understanding of the passage by now, but let's try attacking the question. So does anyone have any questions before we move on?
This may be a little like unrelated, but how do metal ions like magnesium, zinc, and iron help a function of an enzyme?
Yeah, that's a great question. So basically they'll so you know how a protein has like amino acids?
Yeah.
And so it has like side chains.
Yeah.
So the cofactor interacts with the side chains to make sure they're in like the right position for like the reaction to occur.
Oh,
So
so
it
does it change their position, or does it just like structurally like help?
yeah, it does change their position technically. Like it like orients them. So like you know how like charges attract and you have ionic interactions. So it uses those ionic interactions to shift the residues into the proper position so it can catalyze the reaction.
Oh wow.
Yeah. So yeah, that's why cofactors are pretty important. And they're pretty common too. So whenever you see like zinc or iron or magnesium, you usually know those are going to be a cofactor.
Okay, and that's why deficiencies and stuff like that are like pretty serious.
Yeah, exactly. Because we need those enzymes in our body to work for us to be healthy.
Okay, thank you.
Yeah, no problem. So let's go to the questions. Okay. So let's look at what we have here. So first paragraph, we talked about FU, right? And it says FU produces its toxic effects. So cytotoxic, cyto just means like cytoplasmic. So within a cell, it's toxic because it inhibits this enzyme right here that remember it converts nucleotides from one nucleotide to another. So we have a good balance of all of our nucleotides that we need for DNA replication. So that is why 5-FU is toxic, okay? So that's what they're telling us right there. They're just introducing 5-FU, telling us why it's toxic. Then it says, although like many chemotherapeutics, so chemotherapeutics, and it's just another form of saying chemotherapy. So it's it's toxic to cancerous cells, but it's limited in that it can be toxic to the healthy cells, right? It says it's limited by its toxicity and the damage it can cause to the body. So what did the researchers try to do? They tried to develop a chemotherapeutic that's non-toxic to non-cancerous cells. Remember, we want to dodge the healthy cells. So they created an analog of 5-FU, which is called 5-FC, right? We talked about 5-FC. And they made a prodrug. So they made it a prodrug by putting it in conjunction. They joined it with CD, cytosine deaminase, and AB. So CDAB is bound to 5-FC. So that's our prodrug, right? And so why does our prodrug bind to the cell membrane? Like Cam asked before, it's because our AV, free and Extin B, it binds to phosphatidylserine, which is what is on the surface of the cancer cells. So that's how our 5-FC-CD-AV complex binds to the cancer cells, because this AV has an affinity for the cancer cells. Any questions about that? Okay. And so if the CD-AV complex only binds to the cancer cells, localized means like binds to the cancer cells, the 5-FC prodrug, remember prodrug is the fancy name for the entire complex, 5-FC-CD-AV complex. It can be injected intravenously. So intravenously is just into your veins, and it hits your bloodstream. And then so once the 5-FC prodrug is injected into you, it'll go bind to those cancer cells and be converted to its active form via CD, the cytosine deaminase. Remember, we talked about it's the CD within the CD-AV complex that converts the 5-FC to 5-FU. So we want to see if that CD-AV complex actually binds to the cancer cells, right? So then they performed binding and stability assays. So assay, you can just think of that as like another term for like experiment. And so they tested out the binding on 3 different cancerous tissue types: HAE1, MCF7, MDA-MB231. And so they figured out the dissociation constant. We figured out dissociation constant tells us binding between the CD-AV complex and the phosphatidylserine targets, which is like the phospholipids within the cell membrane. So we have the dissociation constants here between our CD-AV and the phosphatidylserine. So which one binds the strongest to the cancer cells based on our chart?
MCF7.
So yeah, exactly, because it has the lowest KD. Okay, then we have our last chart right here. So the data confirmed a strong binding between CD-AV and phosphatidylserine. So we figured out that the CD-AV complex binds to the cell, but now we want to figure out if it actually has an effect on killing the cancer cells. And that's where our immunoblock comes in handy. So they determined the viability of one of the cancer tissue types, so the MCF7 cells. So we're trying to measure the viability of the MCF7 cells at 3, 6, and 9 days post-treatment, right? And so we use 3 different treatment conditions. We use 5-FC only, 5-FU only, or CD-AV and 5-FC. And so it says it was repeated in triplicate. So triplicate means for each condition, we're going to do, let's see, we're going to do 3 trials, or using zero assurance. Think about that for a second. Right. Yeah. So actually, no, they did. So they measured it at 3 different time points. So day 3, day 6, day 9 for each condition. Awesome. So let's look at the question. So I think we already asked this, but so which of the following tissue types does CD-AV bind with the greatest affinity?
MCF7.
Awesome. Yeah, that's correct. Okay. What type of enzyme is thymidylate synthase? So let's look at this diagram right here. What is thymidylate synthase doing to this thing?
Adding a methyl group.
Okay. And what would that make it?
Transferase.
Awesome. Okay. And okay, here it says DNA polymerase, an enzyme necessary for cellular reproduction and tumor formation, requires inorganic zinc to build DNA molecules. Here, zinc is considered what? What would that be?
Cofactor.
Exactly. Yep. Okay. So using fluorescent anti-MCF7 antibodies, researchers immunoblotted MCF cells after 3 days of treatment using 500 micromolar 5-FC Oli, 5-FU Oli, or CDAV plus 5-FC. So what would our assay look like? So basically, what did we say? Bands are going to show up if the cells are viable. So what are we looking at? It says MCF cells after 3 days of treatment using 500 micromolar. So which one is 500 micromolar? We have 0, 500, 1000.
The one in the middle.
Yeah, exactly. And so we're looking at what time point?
3
3
days.
days,
3 days. So which color is 3 days?
Dark.
black.
Yep, dark black. So at 3 days for no CDAV plus 5FC, so 5FC Oli, we have almost 100% viability. So that's a lot, right? We expect the full band to show up. For this CDAV plus 5FC condition, we have 80%, almost like 90% viability. So we would expect the band to show up there too, right? And for our 5-FU Oli condition, we have almost like 85% viability. So most of our cells, MCF7 cells, are still alive. So if the cells are mostly alive for all 3
We should
conditions,
see
what would you-
3 plots.
Oh, which one did you say?
I think D. We should see like all 3 of them plotting. still.
Exactly. Does that make sense to everyone?
Yeah.
At what percentage do you think that we would stop seeing them?
Probably something like pretty low, like around here.
Okay.
It would
20%.
have to be pretty clear. Like
Okay.
they would make it clear somehow. Because of this one, they're all pretty high. And it's and because it's like all or nothing, it's like there's no difference between the thickness of the bands. There's like really you don't really have to differentiate based on this. As long as there's like a pretty high amount, you can just assume it's going to be there. Okay. And last question. Does the information in the passage sufficiently support the researcher's goal? So what was their goal?
It was to create a non-toxic chemotherapeutic alternative to 5-FU.
Exactly. And so did they accomplish their goal of creating a non-toxic drug?
Can I see the table at the bottom?
Oh, yeah, sure. Really 50-50. Or X, yeah. The important part is yes or no. And then once you figure out that. And you can also, for these type of questions, you can look at the reasons why they say yes or no and kind of use that to help you get like pushed in the correct direction.
I was gonna say no, because in D it says the affinity for non-cancerous cells, and that's what they were trying to see.
Yeah, so that is actually right, but it's not the 5-FC that binds to cancer cells, it's the CDAV that binds to it. The 5-FC is bound to the CDAV, but it's actually the CDAV that's binding to the membrane. But the 5-FC is like attached to it. So that is correct. And do you want to kind of give your explanation of how you got to the answer?
Yeah, so they were trying to develop a drug that did not attack non-cancerous cells, So we know that it's gonna attack all cancerous cells. So that's why I was just like A and B is asking about cancerous cells. So I just narrowed it down between C and D.
Right. Because yeah, so they showed that it can bind to cancer cells, but they never tested if it could actually bind, if it was going to bind to the healthy cells, right? The non-cancer cells, because it could bind to the cancer cells, but also bind to the non-cancer cells. We don't really know because they never tested it with non-cancer cells. And so because of that, we don't know if it's not toxic. It could be still toxic. So, and because it's the CDAV complex that binds to the membrane, C would be the correct answer. So does anyone have any questions about that? Any questions? Any questions on the PowerPoint?
I don't think so.
All right. Well, great session today. Does anyone have any feedback? Anything they like to say?
I have a question. How long should reading the passage and answering those 5 questions have taken?
So let's see. So what I like to do is when I'm doing it, I like to take a good amount of time to read the passage and understand it and then go through the questions pretty quickly. So on the MCAT, what I would say is if you can try to answer the question within 30 seconds, if you can't answer, just skip it and move on. And try to go through a quick first pass of the entire section and get as many questions right as you can. So that's my recommended strategy. For the actual passage time, I don't remember exactly, but I can do the math and get back to you next Monday. But or you can just message me. But yeah, I would say is like you want to get a good understanding of the passage. So it's okay to spend some time in the passage, but you want to move through the questions pretty quickly. So that's how I'd go through. And the reason why we take the time to go through the passage now is to make sure that you like understand it. And so in the future, it becomes easier to read them and you'll naturally go faster. So yeah, I'd have to get back to you and do the math.
Okay, thank you.
Yeah, so if no one has any questions, everyone's free to go. I hope to see you next Monday and hopefully we can get some more people too.
Definitely. Thank you, Sid.
Yeah, no problem. Have a good week.
You
Thank you,
too.
Sid.
Thanks, Sid.
Yeah, no problem.